DNA is made up of two strands. Each are complementary and therefore act as a template for building a new strand in replication. In replication, the parent molecule unwinds and the two new daughter strands are built based on base pairing rules. These are the daughter strands of DNA. The nitrogenous bases are paired specifically A to T and C to G. Replication starts at a site called the origin of replication. The two DNA strands are separating, starting a replication bubble. This is replication in a prokaryotic cell. In a eukaryotic cell, there may be several replication bubbles. At the end of each replication bubble is a replication fork. This is where DNA strands are elongating. A helocase is an enzyme that untwists the double helix at the replication fork. Single-strand binding proteins binds to and stabilizes single-stranded DNA until it can be used as a template. Topoisomerase corrects overwinding ahead of replication forks by breaking, swiveling, and rejoining DNA strands. Primase starts an RNA chain and adds nucleotides one at a time using the parental DNA as a template. DNA polymerases catalyze the elongation of new DNA at a replication fork. They add nucleotides only to the free 3' end of a growing strand. Therefore, a new DNA strand can elongate only in the 5' to 3' direction. This is because of the anti-parallel structure of the double helix. Along one template strand of DNA, the DNA polymerase synthesizes a leading strand continuously, moving toward the replication fork. For the lagging strand, DNA polymerase must work in the direction away from the replication fork. It is synthesized as a series of Okazaki fragments which are joined together by DNA ligase. In nucleotide excision repair, a nuclease cuts out and replaces damaged stretches of DNA. Chromosomal DNA molecules have telomeres at the end of their nucleotide sequences. Telomeres do not prevent the shortening of DNA molecules, but they postpone the erosion of genes. Telomerase catalyzes the lengthening of telomeres in germ cells. Telomeres protect cells from cancerous growth by limiting number of cell divisions. There is evidence of telomerase activity in cancer cells, which allow cancer cells to persist. DNA can be damaged by chemicals, radioactive emissions, X-rays, UV light, and certain molecules. Mutations can also occur in which the nucleus is clearly much larger and not visibly enclosed.